This all started last year when I tweeted about this conference that I really wanted to come to PyData, and I asked if there was any money to send me to it, and there was, and so I'm really thankful for NumFocus for giving me this opportunity because I learned so much last year, and I'm really grateful to be able to talk this year. So what I'm going to present is the software that my colleague and I have been working on called Flotilla for open source analysis of large-scale RNA-seq data sets, and specifically we developed it for single-cell analysis. The slides, I just tweeted them. I'm Olga on Twitter, so you should be able to find them. I highly recommend following along and even cloning the repo and running the code yourself there. The repo is olgabot/PyData 2014 biodata. I figured a lot of people's repos are called PyData 2014, so try to make mine a little different. And just so you know, the code is BSD licensed and open source, so please check it out. All right, so what I'm going to talk about today is overall what our design goals were with Flotilla, what we wanted it to do, why single-cell RNA-seq is a really great place to be for data analysis, and I'll show you a few demos of a few features, and then talk about our future work. All right, so the basic idea is that bioinformatics, the processing of data, is not the problem. So the problem is that we don't have enough people who are able to think about the data in meaningful ways and come up with new hypotheses and iterate on them. There's a lot of people who are able to generate the data, but there's not enough people who can do both here. All right. So just to give you a brief history of sequencing, since about 2001, the cost of sequencing per genome has plummeted faster than Moore's law. In particular, now we have cost per genome about $1,000 thanks to this HiSeq X10. And the reason for this, just to show you some biology, the reason for this is that we used to only be able to sequence literally one strand at a time. So you had a strand and you read every single letter on it, and you got this readout for which letter matches. And you see the top line shows only one type of strand, so there's only one read. With the second one, you see that halfway through there's two options, and that's because that's the maternal-paternal versions of that sequence of DNA in that particular person. So this is a very, very low throughput. You can only look at one at a time. Now, using the Illumina HiSeq machines, we can shatter the genome and read many different sequences at once. And they have this great new product, HiSeq X10, which is great for genomic sequencing as well. All right. There's a lot of pipelines to take this huge amount of data and get gene expression values, or like what genes are being expressed in cell, or what mutations are around in a particular person or a particular sample. But there's not that, but it's really difficult to get, so okay, fine, I have these genes, but what does that mean biologically? What does that mean in terms of what is happening in the cell? What does that mean for my biological question? So what we wanted to create was a platform for data-driven conversations about biology. So we want to promote interactive data exploration. So unlike the previous presentation, I won't have any fancy, shiny JavaScript plots. I just have static PNGs, but we'd like to go into that direction. We also want to decrease the time for iterating over hypotheses, so hence the widgets help a lot. So I work with a lot of biologists, and we want them to do the biology. So what's great is that now with Flotilla, I can give them the code, I can give them the data, and say, here's what you can look at, and they're able to explore on their own genes that they care about and systems that we care about. Using the IPython notebook, we can promote reproducibility. It's still kind of up to the, it's not complete data provenance, but we would really like to get to complete reproducibility of data sets. And what's great about this is that now when I meet with someone, I show them a result, I can say, okay, well, here's a result, and they say, oh, well, what if you did that only on this subset of genes, or you only looked at these samples? And now in meetings, I can live code and show them the result, and that has been really, really great and really accelerated our discovery process. Now, how to get Flotilla? So we have a few different ways. So we have a Docker instance on our documentation. You can see how to run it if you already have Docker installed. This is the easiest way, most reproducible. You don't have to install any of the dependencies, don't have to worry about anything. You can also install directly from PyPy. I just pushed a release like an hour ago, so you can check it out. You'll need the latest release to follow along with this presentation. And also on GitHub, we'd love for you to contribute with your awesome features. All right. I really like this octocat. All right, so what is single-cell RNA-seq? What does this even mean? So you've probably heard of genome sequencing. You may not have heard of transcriptome sequencing. So the transcriptome is what genes are currently being expressed in a cell. So the genes that are being used in your skin cells are different from your heart cells, are different from someone else's skin cells. This is a very specific signature of what genes are being used. And the transcriptome is specifically the RNA molecules that exist in the cell. So DNA is a very stable, good blueprint and book to read from, but it's not that portable within the cell, so you copy the DNA to the RNA molecules, and then these can be used to travel as messengers of information throughout the cell. Now, why single-cell RNA-seq? So this is one single cell. You may wonder why we care about this one, just one of them at a time. And the reason is that these are pretty cool. This is not, it's just a smooth cell. There's a lot of things going on. It's kind of hairy. There's a lot of genes being expressed that are different from other cells, and they're all unique. So what I think is really fascinating is that if you take a pooled sample, you have some value for gene expression. So for example, what is the average eye color of people who live in New York City? You get a value, right? You don't get the distribution of what our eye color is in New York City and how that compares to eye colors in other cities like Atlanta or Tokyo. You only get one value. So from single-cell RNA-seq, you get to see this whole distribution of gene expression across populations. All right. Now, how does this actually work? A few years ago, this company called Fluidigm created a single-cell microfluidic chip that allows you to sort individual cells. So that's what one of these little chips is. And if you're curious about how it works, you have a suspension of cells in liquid. You have to have a pretty high density, so about 250,000 cells per milliliter, and then you shove them through this microfluidic chip, and they stochastically sort into these cells. And there's 96 cells, so you have to have a lot higher density than as cells that you get in. And you don't get 100% capture, as in you don't always get 96 cells every time. It kind of depends on your cell stickiness. So if your cells are really hairy and have a lot of things sticking out of them, like neurons, they can be a lot harder, but a lot of blood cells are easier. All right, so on to the demonstration. If you're following along, I'm going to use this data set from a paper published last year on single-cell immune cells in mice. So what they did is that they took bone marrow-derived immune cells. So bone marrow are the B cells, and of the immune cells, the T cells are ones derived from the thymus. So B is for bone. And they subjected them to an immune insult, which is lipopolysaccharide, which is I don't even know what it does. This is not my field. I don't study immunology, but it does some kind of, it simulates an immune insult, such as if you have an infection or some other inflammation. They have 18 cells, so that's relatively small in the world of single cell at this point. The same authors in the same group published a paper this year with almost 2,000 cells. So this is much smaller, but a nice data set for this demo. And as a side note, I have cleaned up this data and reproduced all the figures from their paper in a recent blog post. So I would encourage you to check it out if you're curious about how this data was actually created. All right, so let's get started. So the data is saved to your home directory / flotilla projects, and then the name of the data package. All right, so we've downloaded a bunch of information. We get some progress. We get a few error messages just saying that I didn't provide any colors and that we didn't remove any samples that had too few mapped reads, meaning that they weren't sequenced deeply enough to really trust them. All right, in case you're curious, all the study object is just a wrapper for holding data frames together. So study.metadata, the metadata object has the attribute .data, which is a pandas data frame. We tried for a long time to subclass the pandas data frame and learned that that is the wrong way to do things. Even the pandas developers say that you should just make a new object that has a data frame attribute. It becomes a lot easier to work with. It's a very powerful tool. There's a lot of things going under the hood, so I'd recommend just to let it do its own work and then you do yours separately. So metadata, as you can imagine, it says what is the phenotype of these different cells. I've already filter them for mature and immature bone marrow-derived dendritic cells. And whether or not this sample was a pooled sample, so instead of a single cell, it's about 10,000 cells, and whether it's an outlier or not. Then the expression data, so our data classes follow the scikit-learn standard of the samples are the feature name, or the samples are the row names, and the features are the column names, because we make heavy use of scikit-learn, so we want to stay consistent with that. And what's nice is that most of the heavy lifting in our package is done in this base data object, and that has all kinds of great functionality like dimensionality reduction. So if you make a new object that if you make a new data type, that inherits from base data, it will already have a classifier, it will already have a dimensionality reduction, it will already be able to standardize itself. There's a lot of great functionality already been built in. So while we were building this, we realized that we don't just have a model view controller as in other REPL or other design systems, we also have this backend computer. So we wanted to build in, make sure that we nicely build in our compute backend. And our viewer, we would certainly like to use a more JavaScripty interface, but for now we have this PNGs. And what's nice is that with this compute class, we can really easily create new decomposition algorithms. For example, this data frame reducer base abstracts out the scikit-learn and what things we specifically like to do with it. And then you can create this new object with very little code, which is really nice. All right, so let's get to the demo. So questions that we're going to ask are doing PCA on different data types. So with this function, I can now get the PCA output from this data. If you've used scikit-learn in the past, you may know that getting the information out is quite easy. It's very nicely designed, but getting it plotted in the way that you want can be pretty tricky. So with a single command, we're able to do this. So now we can try doing this on different data types. So the splicing data is not quite as clean. You'll see that there's these really bizarre, crazy ideas about where this thing is located in the genome. So let's go back to expression data. And I should point out that the feature subset we're using that's called variant is our default feature subset because it squishes down the number of genes you're looking at a time. What it is that the variance of these genes is at least 2 standard deviations away from the average variance of all genes. All right, so we're back to gene expression. Let's look at just the samples that are not pooled, just the single cells. Right, so now these orange ones are from the paper. They found these to be more mature. And let's look at what happens if we do this feature-wise. So now what this transposes the matrix. Every dot in here is now a feature, it's a gene. And the loadings are now the samples. So let's look at this. So this S12, 16, and 13 seem to be on the far right side of principal component 1. Let's go back. Not feature-wise, and let's look at these genes that are involved that they curated that are known to be involved in the lipopolysaccharide response. All right, so this is actually the exact transpose on the x and y. The x-axis is flipped on the negative, but that's just a matter of initialization. And this is basically exactly the figure from the paper, which is really cool. And now what we can do is, so we have these mature samples, and again, this would be really great to have a hover object, but we don't have that yet. So these orange ones, I know, I apologize if you can't see it, but they are S12, S13, and S16 as we saw before. So this allows you to look at your data in multiple different ways. Finally, a lot of times we'll ask, these PCA plots can be hard to interpret for the first time for someone seeing them, so they'll see a vector going in some direction and may not necessarily know intuitively what that means. So let's look for this gene called SLC7A11. So we have, we'll finish up with these violin plots. All right, here we go. So we plot all these violins. So let's look for, this is alphabetical, so let's go to SLC A11, and that is here. So we saw that, if you remember correctly, we saw that its direction was greater towards the green samples and away from the orange samples. So now you can start creating a mental model of what do these vectors actually mean, and seeing that, well, if it's in this direction, it's more highly expressed here. So this can be really, really great. Oh, and I should mention that these black dots are the pooled samples. So the pooled samples do not always exactly represent the single cell. So this just further emphasizes that. So this can be a really great mental model for especially the biologists I work with, because the PCA, it can make sense, but in terms of what happens to the genes and the expression itself can be not quite intuitive. All right, so let's now, one other thing I want to show is that you can, so let's turn off the violins because they take a little bit long to plot. And now you can also, what we often get asked is, can you do this on my favorite gene list? So we can input a specific list. This is just a link to a Dropbox file. It looks like a text file, as you would expect. So a bunch of weird gene names. And now we can perform, so we can't just use this straight. We have to say that we want this custom gene list. And wait a little bit, and now we have our PCA calculated on this specific gene list. I chose it to be the LPS treatment, but you can choose to be anything you want. So in our lab, we often study neurodegenerative diseases. So someone has a list of their favorite autism-related genes or their favorite ALS-related genes, and they'll use them. All right, so now we can start looking at networks. So this, from the default parameters, what you see is that we're trying to draw let me go back actually to these things. So what we do first is we reduce the dimensionality of the data. That's a big part of what we do first. Reduce it, then using the first few components, calculate covariances between samples, just using those five dimensions. Use a cutoff on those covariances to decide whether or not these samples are related enough that we want to draw an edge. Use a degree cutoff for whether or not we want to plot that sample or not, because if it's a loner, we don't want to plot it. And a weight function for the edges to push the samples farther apart. And finally, a quote-unquote heat map of the gene of interest. So let me show you that. So we have this graph. It's not too connected, so I would like to see how more samples are, more samples and how they're connected. So let's decrease the cut of the covariance. So this is a plot of overall covariance, and this is where our cutoff is. So if we cut off more to the left, then we'll have more samples and we'll have more connectivity. All right, and this is so we try to have some informative plots about what your plot overall looks like. We can increase the number of components to 11 because 10 is too little, and right. Now, one gene in the paper that they emphasize is this gene called IRF7. So if you, IFR7. All right. All right, well, I'll skip that for now. So what is also nice is that, again, you can draw the labels if you like, and keep interacting with this plot in many different ways. All right, so next we can do a classification between samples. So this doesn't work at first because I'm trying to classify on this outlier variable, which doesn't exist in this data set. So let's classify on the maturity of these cells. So as you saw, this is going to be the orange versus the green before, and let's use not the housekeeping, the LPS response genes, and let's look at, again, not the pooled samples, just the single cells. All right, so this is going to calculate this, and now this is plotting just the top 23 features and doing PCA on just those top 23 features of the LPS response that discriminated between these two categories. And what's interesting is that we come up with actually a few different genes than what they came up with in the paper, so we can look at some more new biology here. All right, so moving on to a clustered heat map, as of Seaborn 0.5.0, I helped create the clustered heat map in this package, and now you can easily create clustered heat maps using SNS Seaborn cluster map. And what's nice is that you can, because it's really not trivial to do in Python. In R and MATLAB, there's great libraries for it, but as a Python evangelist, I felt like it was my duty to do this. So with this cluster map, what you can do is, so this is a clustered heat map of these samples. So again, the rows are the samples and the columns are the data. Now we can take this and let's look at the LPS response genes. So you see these orange samples clustered together again. All right. And then we can look again and see what kinds of genes are clustering together with these samples, and their dendrogram is far away, so they are actually quite different from the remaining cells. And yeah, so do you see the streak in the middle here? That's the pooled samples. So they are way more similar to each other than they are to the single cells because they have this big average of everything. Right, now, and then we can also play around with different metrics. Let's try, since we're in Manhattan, let's try the Manhattan city block distance. And we can also use different linkage methods such as ward or complete. Don't use single, it's not a good one. And let's also, again, do not the pooled samples. So again, this takes a little bit to compute, so the pooled samples are still up here, and it's still calculating from the black dot, so let's wait a little bit. And again, all right, cool. So now we can see which gene clusters are actually contributing here. All right, next, this last one I'd like to show you is this interactive correlation. So what you can do is calculate correlations between samples or between features. So again, this is a little bit more subtle than the previous one. What I want to emphasize is that if you change, for example, the feature subset of this, what you're saying is that now I only care about the correlations between these features between these samples on this feature subset. So it's a little subtle there. And again, you see how these pooled samples are way more similar and how these S16, S13, and 12 all keep clustering together. So this is many different ways of viewing the same thing, that these three samples are really different. And this allows you to explore your data and find different ways of showing, of proving the same point. All right, let's also try, feature-wise. This may take a little bit of time to calculate this. But it's pretty exciting. So this feature I literally released this morning for Flotilla, and it's been really exciting to be able to work with it. Right, I think I'm almost out of time, so let me just end with this. So I would love to see many of you contribute to Flotilla. I see Flotilla as a gateway drug. Ooh, cool. So this is the correlation between all genes, all lipopolysaccharide response genes in our data set. And again, you can always save this figure as some file that you like. All right, so I would love to see you contribute to Flotilla. I personally see it as a gateway drug for biologists into coding because as you saw, within a few commands, you can do really powerful things. But research is not one size fits all. There's no way we could ever possibly come up with every possible analysis that someone wants to do. That's why we've kept the code, the underlying code and the underlying data frame always around, so you can always do whatever analysis you'd like to do. And as you notice, all the data needs to be is in this scikit-learn format of features by samples. It's just a data matrix. So any non-biological data is also welcomed. All right, and I would like to thank the rest of the lab, Jean Yo, especially my boss, Mike Lofsey, who pioneered the work of the interactive widgets and presented this similar work at a biological data science talk. Yan Song, who's right here. So she performed all these like scary wet lab experiments with cells and stuff. And before we used Travis CI, there was Yan CI who would come up to me and say, Olga, this is broken now. I'd be like, yeah, probably. I'll try to fix that. And the rest of our single cell team, what I'd also like to thank funding from Gnome Focus. I'm a John Hunter Technology Fellow, and my graduate fellowship and UCSD Bioinformatics Systems Bio.